Hey, welcome everyone. It's my great pleasure to introduce the Vivaldi webinar on yeast fermentation. Let me start with a very brief overview of um, a bit of background about Vivaldi and the webinar. So first of all, my name is Dieter Matanovic. I'm one of the PIs in the Vivaldi project, and this should not happen now. Um, the Vivaldi project is a European project funded under the Horizon 2020 scheme with the mission of turning CO2 emissions into sustainable bioproducts. And as a very brief overview, this is the map of partners. So you see that we are really like spanning all over Europe here with our partnership. And as a very uh, uh, basic and general overview of the mission, Vivaldi is working on industrial, let's say, byproducts, or what we may call waste streams, and valorizing them. On one side, gas emissions, and there we are mainly focusing on CO2 as a resource, and on the other side, on wastewaters that contain valuable nutrients for biotechnology, as you see, like ammonium and other cations. On the CO2 stream, the mission that we have is to purify and concentrate gas emissions, CO2 from gas emissions, and turn it into valuable carbon sources for biotechnology, mainly formic acid, and the other aim also is on methanol using electrocatalysis. And also the nutrient recovery is based on essentially on electrochemistry. And both streams then are dedicated to go into fermentation, where we use a methylotrophic yeast, Komagataella phaffii or Pichia pastoris, as you may know, to produce organic acids that are valuable base chemicals. And you see the list of acids on the right side: 3-hydroxypropionic acid, lactic acid, succinic, and itaconic acid. And you will hear about the progress on using this yeast in fermentation to produce these different acids. So a brief overview of the program. So we will start with Professor Pau Ferrer from the Autonomous University Barcelona, telling us about the production of 3-hydroxypropionic acid with Komagataella phaffii. Then Dr. Simone Bachleitner from Boku University is working together with me on lactic acid and itaconic acid, and will update you on the progress and quite remarkable achievements that we have in all these fields. And I'm very happy then to also introduce Professor Valeria Mappelli from the University Milano Bicocca as a guest speaker here, who will further extend the scope towards other resources that are considered as waste now. And Valeria will tell us about the valorization of cotton waste with yeast engineering and fermentation. So we have these three talks first, and then an open discussion, Q&A. This will work in the way that you will be able to type your questions into the Q&A. So use the Q&A button in Zoom, type your questions. You also have the option to like questions so that they might be ranked higher to be answered. And then I will moderate that and read the questions and hand over to the speakers to answer the questions. So let's get into it. I'm very excited about the 3 talks to follow, and hopefully a very lively discussion session. And yeah, I hand over to Paul now for the first talk. Thanks everyone to be here, and yeah, looking forward to the webinar. Paul.
Yeah, thank you very much, Dieter. We'll wait till okay, there it comes. So yeah, so thank you very much for the introduction. And my talk today will focus on the work we've been doing on developing fermentation strategies to produce 3HP, 3-hydroxypropionic acid from methanol, and we will see some work also using mixes of methanol and formic acid. Okay, next slide, please. Yeah. So first question is why methanol? Methanol, it's a highly reduced one carbon feedstock. Okay, it means that it's very attractive to produce a number of wide range of products with different degrees of reduction. Okay, and it's also more reduced than, for instance, other standard feedstocks like glucose or glycerol, for instance. And on top of that, methanol you can see here on the top in this figure that it can also generate NADH and subsequently energy from direct oxidation of methanol to CO2. So altogether, it's an emerging, has a potential to be an emerging biotechnological substrate for bioproduction of metabolites. So regarding the product, 3-HP, it's included in the top 10 list of value-added chemicals from the Department of Energy in the US. And you can see that you can produce wide range of products from acrylic acid and acrylamide, 1,3-propanediol, and even polymers. Next slide. Yeah. So in nature, you have several organism bacteria that can produce naturally 3-HP. In yeast, either as a final product or as an intermediate in several pathways. So in yeast, essentially, they have been engineered essentially with two pathways. One is the malonyl-CoA reductase pathway, which takes one enzyme from a couple of CO2 fixing cycles described in archaean bacteria, and it has been tested in several years. So essentially, it's an enzyme, bifunctional enzyme that takes malonyl-CoA. It comes from pyruvate, and it can be reduced to 3-hydroxypropionic acid by using NADPH. Next slide, please. And the other alternative that it has been also tested in yeast is the so-called beta. It's a synthetic pathway, so the so-called beta-alanine pathway. Okay, that it was designed by a group in Irina Bonordina in DTU. Okay, it essentially uses 3 different enzymes from different organisms, also requiring NADPH to produce 3-HP from pyruvate. Okay, next slide. So in our work, we decided to go for this, to introduce the synthetic beta-alanine pathway in Pichia, essentially because the theoretical yield on methanol is slightly higher than for the malonyl-CoA pathway. Okay, and all the genes were cloned under the control of methanol-related promoters. We've done some additional strain engineering. We'll come, for instance, introducing formic acid dehydrogenase depending on NADPH to sort of balance the redox. So sorry, to balance the NADPH balance. And we'll come to that later on. Next slide. So when we started, we actually didn't have much knowledge on zero knowledge, I would say, on producing organic acids at bioreactor scale with Pichia. So we took advantage of the fermentation technology that we had in place for for recombinant protein production. As you know, Komagataella phaffii has been essentially used for protein production over the years, for 30 years, maybe 20, 30 years. So the standard fed batch scheme, it's shown here. It has a first growth phase on glycerol, okay, where you essentially generate biomass, and then you have a transition phase where you can have a limited methanol glycerol feed and also methanol, where you adapt cells to methanol, and then you have a methanol feed phase for production, okay, where you can test different alternatives that you have here, okay. So you can add different pulses manually or automatically. You can have constant feeding rate, okay, so you will have a decreasing growth rate over time, or you can think on an open loop pre-programmed exponential feed rate, okay, to reach a constant growth rate in the reactor. Okay, so this one is the strategy that we initially implemented. So in the next slide. So for this, we first had to decide on which was, or select the optimal growth rate for this exponential feeding rate, so to achieve constant growth rate. Okay, and for this, a nice tool to select for the best growth rate, it's the use of chemostat cultures, where you essentially feed in fresh medium constantly, and then you remove an equal volume of fermentation broth. And for this, you can control growth rate, essentially manipulating the feeding rate. So the dilution rate is equal to growth rate. Okay, so here it's a series of experiments we ran of chemostat cultures at different dilution rates, and you see that the optimal conditions in terms of yields and productivities and space-time yields is the range of 0.03, 0.35 hours minus 1. So that's the optimum growth rate range. Okay. So next, in the next slide, we decided to implement this strategy of feeding methanol feeding, exponential feeding to achieve this growth rate, and we use that to just, for instance, characterize different strains having different genetic constructions. Okay, so this we did. You can do it at 1 liter scale. Okay, very easy. And from there you can calculate all the growth parameters, and you can see, for instance, the strain on the right hand side. You have more or less the same production titer, final concentration, but it has slightly less biomass. So if you click again, so it means that you produce the same biomass, same product with less biomass. And this, if you click again, yeah. So you can see that essentially the productivity, the specific productivity and the specific consumption, methanol consumption rates are improved in the right strain. Okay. So the next step was to improve. So this is one application to really screen and characterize strains that you construct in the lab. But of course, we wanted to improve further the conditions. So we moved to 5 liter scale and implemented the same strategy, but a longer time. And here we ran into surprises that, for instance, you can see that in pink, in cyan, you have the residual methanol concentration in the reactor, and you can see that after 40 hours, methanol starts accumulating, which it shouldn't in principle. Okay, and you can see that the reduction in growth rate, okay, and production of 3HP is clearly shown. Okay, this problem we couldn't solve it by just reducing growth rate. Okay, so after 40 hours, so we had to think on something else. So next slide. Okay, so the next strategy was to implement a methanol closed loop control. So controlling methanol concentration based on a methanol probe and monitoring methanol concentration in the reactor online in real time. and based on that, correct the feeding rate. Okay, and you can see that in using this strategy, you can actually control your methanol. You can keep your residual methanol concentration in the reactor down to 2 grams per liter, which was the pre-selected set point. And the way to do this is, you can see here on the left-hand side that the flow rate, it's not exponential anymore. So it goes, is the orange curve. It shows it's not exponential, so it slows down. So it sort of flattens towards the second part of the fermentation, and the result of that is that the growth rate, it's not constant anymore, or the pressure, so it goes, also it gets reduced. Okay, so by this you could actually improve a little bit production, and you can see also how this is done. So actually, if you look at the right hand side, the QS profile, so the specific consumption rate of methanol, it goes, what we do, it's reducing that, and with that we sort of slow down the growth rate over time. So if you go to the left hand side, you see that we managed to slow down the growth rate much earlier and to avoid this accumulation of methanol. Okay, so next slide, please. Okay, so so that was one way of improving the performance of our cultivation. The next objective was to investigate whether we could improve performance by co-feeding methanol together with formic acid. Okay, so in principle, Komagataella is not able to use formic acid as a carbon source, although it has the potential, the metabolic potential through the reductive glycine pathway. Okay, but growth cannot be sustained. But still, remember that you can fully oxidize formic acid to CO2 again and get some extra NADH. And with this strain we generated, we hoped that we could have generated some extra NADPH as well. Okay, and in fact, some preliminary experiments done in shake flask, okay, by some other members of the project in our department, by Zainab, okay, managed to improve like 15-20% yield of 3HP by doing this co-feeding of methanol and formic acid in shake flask. Okay, so next, so first thing we thought, so it was to investigate which relation of methanol and formic acid should we use, or should be the optimum for 3HP production. Okay, and to do that we took advantage again of chemostat cultures. Okay, and the first thing we notice is that when adding sodium formate in the reactor, you see a very sharp increase in the pH. Okay, so in the left graphic, you see the control strain, so wild type strain producing no 3HP. So you start with pure methanol, and as soon as you add formic acid formate, you have a sharp increase on pH. Why? Because we only control pH by adding ammonia. Okay, so here we don't have a acid solution to lower the pH. Okay, when the reactor pH goes up. Okay, so you can see also that you have a graph where you have pH. It says pH control. This means that by adding hydrochloric acid, so having both ammonia and hydrochloric acid as pH control solutions, you can really manage to control pH at the set point. With the producing strain, so it's the right-hand side, we thought maybe you don't need hydrochloric acid to control pH, so keep it at 5 because you have 3HP production. But not even with this, you manage to control pH at 5. So you still have this increase. Okay. So in the next slide, you have what happens with the wild type strain. When you co-feed in equimolar amounts methanol and sodium formate, effectively you're able to increase at different extent, but you're able to increase the biomass yield by 10, 15%, 10, 20%, depending on whether you keep pH control at 5, which is a standard pH, or you leave it free, meaning that it goes up to, as soon as you put formate, it goes up to pH 7, for instance, okay? So then we went up to see what happened with the production strain, and here you can only see some. Can you click again, please, Lara? Yeah, you can only see a slight increase in let's say in specific productivity and product yield on methanol when you leave the pH free. Okay, if you keep the pH at 5, you don't see any difference. Okay, so there's a big impact on the pH, on the process metrics, on the performance. Okay, of course, if you consider yield on total carbon use, because of format as it's burned to let's say essentially burning to CO2, the overall yield based on carbon mole yield it goes down. Okay. So next slide. Yeah. So clearly, continuous feeding of co-feeding of sodium formate in the fed batch, we thought it would be really challenging. So we went for let's say an adaptation of what Zainab had done in shake flask, okay, and also tried in repeated batch experiments in bioreactors, which was essentially going back to this strategy with a methanol closed loop control, where we keep methanol residual concentration at 2 grams per liter, and then manually add some sodium formate pulses. Okay, you can see on the left what is the impact of result of adding a methanol pulse. So in green you see the sharp increase in pH every time you make a pulse, and the sharp increase on CO2 production just after the formate pulse, which is the brown, the dark curve. Okay, and with this system, so in between, so you see that pH goes down again. This means that formate it's consumed again. So you leave a little bit time, and then you add another pulse of formate, and so on. You repeat it over time, and what you see here, I will see you in the next slide, that you effectively are able to increase process performance. Okay, and essentially you are able to keep the growth rate mean decrease to a minimum, so you can sustain growth in a better way. Okay, and also the methanol specific consumption rate, it's also kept sustained for a longer time. Okay, and with this, of course, you improve productivity of the system. Okay, of the fermentation. I think in the next slide you see an overall. If you click 2 or 3 times, you will, Lara, some arrows will come up. Yeah, you see essentially 3 fermentation. So the 3 fermentation strategies. So the pre-program experiential feeding of methanol, okay, to 2 different runs. The second, so 2 other runs with methanol closed control loop. Okay, and finally the last line, it's the closed control loop with formate pulses. And you can see, for instance, that the final 3 HP concentration in the supernatant goes up to 50, from 33 grams per liter to 48 grams per liter. Per liter of reactor volume, it's obviously lower because you have the biomass, so you reach 38 grams per liter in the best of the cases. And I clearly have an improvement, right? Combining methanol control plus formate pulses, you obviously have an improvement on both space-time yield and product yield. Okay, So next slide. Yeah. Finally, these are the two final slides. What can be, so we have improved somehow our fermentation capacity or performance. But still there's quite a lot of room for improvement. We are still far from our theoretical yield on methanol in terms of product yield, okay? So we've done some additional strain engineering work, essentially focusing on overexpressing genes encoding for lactate, Y lactate, because it's very close. It's an isomer of 3HP. So here, very briefly, I can show you some results very quickly on how we can improve just by overexpressing one of these transports, where we can improve up to 48% 3HP in final concentration, and 20% space-time yield is also improved when you overexpress this transporter. Okay, and more interesting, it's that this strain overexpressing the transporter, it's more robust at lower pH. I think in the next talk, we will want this pH issue will come again. Okay, essentially you save ammonia for pH control. Okay, so here you can see essentially how all the metrics are improved just by overexpressing this transporter. And very interestingly, both in terms of product yield, okay, so you go from 0.15 to 0.19, almost 0.2 gram based on methanol. And also very interesting, it's the yield on CO2, which it's lowered. Okay, so we go from 0.8 to almost 0.7, okay, grams of CO2 per gram of methanol. So it means that by introducing this transporter, you are able to manipulate the split ratio between methanol full oxidation to CO2 and methanol assimilation into biomass in product. Okay, next slide. Okay, so these are my final remarks. Okay, I think we can prove that we can adapt conventional fed batch strategies originally designed for protein production, or you can adapt them for organic acid production. Okay, and by doing this, we show the potential of this cell factory for bioconversion of methyl into chemicals. Of course, we need to do further process optimization, adapting growth medium to solving some of the problems we see, for instance, with pH control, where I haven't mentioned it, but we end up with an excess of ammonia in the fermenters because we generate a lot of acid, so we need to add more ammonia than needed for growth and product formation. Okay, and clearly we need a process where we avoid the use of glycerol. Essentially, we only want to use methanol. Okay, and yeah, further work on formate co-feeding. Okay, could be done. It's not optimized what I show you. Okay, and then of course this needs to be combined with further strain engineering. Okay, and with this, I think the next slide is just show contributors of this work. That okay, Silvia did all the strain construction, Felicitas and Giovanni did all the fermentation work with chemostat and fed batch, and now work on the control, developing the algorithm for control of methanol, and Zainab did all the small scale work with formate co-feeding. Okay, so thank you very much for your listening, and these are the contact details.
Thank you, Paul. So yeah, that was very exciting. Before we continue, you can type your questions already now into the Q&A. So just go to the bottom line. They will be answered at the end. But if any question pops up, just type it in so we have it saved there. Yeah, it's my pleasure now to introduce Simone as the next speaker, Simone Bach-Leitner. And Simone will tell you about lactic acid and itaconic acid, mainly, so work that has happened in our lab over the last 3 years. And yeah, Simone, the stage is yours.
Thank you, Dieter. Okay. So I'm going to talk today about the opportunities and challenges of yeast-based lactic and itaconic acid production when we want to use methanol as a carbon source. And as we've heard before, organic acids are quite valuable for the industry because they have quite diverse chemical properties and can be used in a wide range of applications. And we are interested in producing lactic acid, which is a hydroxylated chiral carboxylic acid. And it is mainly used in the chemical industry, but also in the food industry, in the pharmaceutical industry, and in the recent years also more and more in the polymer industry, because it can be used to produce a polylactic acid, which is kind of a sustainable and compostable form of plastic. The market price is quite high with 4.9 billion by 2026. And in industry, lactic acid bacteria are used to produce this organic acid. And here titers are reached between 100 to 200 grams per liter when glucose is used as a carbon source. And the other acid that we are interested in is itaconic acid. It's kind of a newcomer. It's an unsaturated decarboxylic acid, and it has been shown that it can be also used as a building block to replace petroleum-based polyacrylic acids. So you can use it to form absorbents, polyesters, also plastics and synthetic fibers. The estimated market price is lower than compared to lactic acid, and it can be produced, or what industry is using is filamentous fungi to produce this itaconic acid, for example, with Aspergillus cereus. And here, titers between 90 to 100 grams per liter are reached. So I won't go into detail with the yeast that we are using to produce our organic acids, because we have heard already about it in Pau's talk. I just want to mention that as Komagataella phaffii is a natural methylotroph, it's really the perfect host to produce these organic acids. And as it is already so established in industry, it's the tools that we need to work with it are there. So now let's dive right into lactic acid production with Komagataella phaffii. Here I would like to mention Lisa, Gregor, and Rosanna. All three of them have been contributing to this story a lot. So when we want to engineer now a yeast that is capable to produce lactic acid, we need to provide it with an enzyme, a lactate dehydrogenase, that can convert the pyruvate to lactic acid. And we have been choosing an LDHL from Lactoplantibacillus plantarum. We put it under a very strong methanol-inducible promoter. And as you can see here in a first shake flask cultivation, this LDHL strain is now capable to produce lactic acid. However, the titers are really low, and after some time, the yeast starts to consume the lactic acid again. In literature, there is another enzyme described, cytochrome B2. It's an L-lactate cytochrome C oxidoreductase, and it can also convert the lactic acid back to pyruvate. So when we delete this gene encoding for cytochrome B2, it was possible to boost the lactic acid titers a bit. However, it didn't prevent the consumption. And this is mainly due to the fact that our LDH enzyme is working in bidirectional ways. So we were curious if this is also the case when we run a bioreactor experiment with this strain. And as you've heard already in Pau's talk, we are working with those different phases. We have initially a batch phase for biomass accumulation, followed by a glycerol feed phase, which is important to induce the methanol genes. And then we have a long methanol feed, which is basically our production phase. As in this phase, the yeast starts to produce lactic acid because we're using methanol-inducible promoters here. What you can see here is that we generate quite a lot of biomass. In the end, we end up with 140 grams per liters, and we only produce about 4 grams per liters of lactic acid. We have been testing 2 different temperatures here, but they don't really make a difference. And these 4 grams per liters that we reach here are similar to other systems that have been tried for lactic acid production on methanol. For example, another group wanted to produce D-lactic acid using Komagataella phaffii, and they can produce up to 3.48 grams per liters. And also Komagataella polymorpha was engineered for lactic acid production, reaching also around 3.8 grams per liters. So the titers that we achieve here are very similar to what is published. However, we were really astonished by the fact that our yeast starts to consume the lactic acid, although methanol is present as a carbon source. So we performed another shake flask cultivation where we took our wild type strain and we grew it in a methanol feeding phase, basically. And then after some time, we added lactic acid to our cultures. And we had 2 sampling points, one in the methanol feeding phase and the other one after the addition of lactate. And then when we had a look at the gene expression of metabolite methanol utilizing genes that are important for the methanol pathway. That are AOX1, FDH1, and DAS2. And what you can see here very nicely is that when methanol is present as a carbon source, the genes are quite active. But as soon as we add our lactic acid to our methanol cultures, the gene transcription gets reduced quite a lot. So it seems that our product, lactic acid, affects the transcription or the gene expression of the methanol pathway. And in literature, we know there are some transcriptional regulators described for the mood genes. We have here some transcriptional activators like MIT1 and MXR1, and also transcriptional repressors, MIG1, MIG2, and energy1, that play kind of a crucial role when also other carbon sources like glucose are present in your culture. So we have been investigating now these transcription factors in our strain. That means we have been deleting the repressors. And on the other hand, we have been overexpressing these transcriptional activators. And we have been also generating a strain where we have been overexpressing both transcriptional activators. Then we screened our strains again in a methanol shake flask cultivation. And what became apparent is that when we overexpress these activators, the strains, they are quite reduced in their growth phenotype. So the overexpression of MXR1, but even more the overexpression of MIT1 together with MXR1 leads really to a decreased growth of our strain. However, these were the strains that produced best. So here we could really reach higher titers of lactic acid. So we took now this best performing strain with the overexpression of both transcription activators, and we performed a bioreactor cultivation, a fed batch cultivation with it. We again have the different phases, and what we can see here also in this bioreactor cultivation is that our new overexpression strain grows not as good as the parent strain, but it produces more lactic acid. So here we reach about 20 grams per liter after 92 hours. However, when the fermentation is continued, the cells start to consume a bit of the lactic acid again. So it decreases to 15 grams per liter. And when we had a look at the gene expression of the methanol utilizing genes throughout the fermentation process, we see that in our overexpression strain, the expression of the methanol utilizing gene is, of course, strongly increased in the beginning of the fermentation phase. But then at the end of the fermentation phase, it reaches a similar level as in the parent strain. So we believe that this lactate consumption consumption at the end of the fermentation might correlate also with the weakening of the expression of the methanol utilizing genes. So what we can see or say now is that lactate as a product on methanol is definitely challenging. And there are some regulatory traits that we still need to discover to improve lactic acid production on methanol further. So the state of the art in industry, as I've mentioned before, are between 150 to 200 grams per liter when you use lactic acid bacteria using glucose as a carbon source. And we are still quite far away from it to be here in a compatible range. The story looked more promising with itaconic acid. So here I would like to mention Manja Molgaard-Severinsen. She has been engineering a strain that is capable to produce high amounts of itaconic acid. And to do so, you need to provide the yeast with three enzymes. So the key enzyme is basically cis-aconitate decarboxylase, which decarboxylates the cis-aconitate to itaconate in the cytosol. And then you also need two transporters, one mitochondrial one, MTTA, and one exporter, MFSA. And in a first bioreactor cultivation, you can really see here very nicely when you have a strain expressing only CAD-A and MTTA, you can reach up to 22.9 grams per liter of itaconic acid. But when you express all three of the important genes, with the addition of the MFSA exporter, you could increase the titers to 28.2 grams per liter. And these are already quite nice titers to start with. A very common strategy now to improve the titers even further is to generate strains that harbor multiple gene copies of the itaconic acid pathway. So here in black, you see our single copy strain, and in colors, blue, gray, and violet, we have now multi-copy strains. And these strains, they have more copies from CAD-A, MTTA, and MFSA. And the best producing strain was actually MSC1, which produced almost 50 grams per liter of itaconic acid in our methanol fed batch cultivation. And what you can see also very nicely here is that the gene copy number correlates with the gene expression of the itaconic acid genes. In Vivaldi, it is not only important to produce now a very high producer strain, but also to develop a process that is sustainable. And we had the luxury to work very closely together with our downstream processing partners, Processium, and they basically provided us with feedback how we can optimize the process so that we make it possible for an efficient and also cheap downstream process. Here in F1, you see now this high producing strain in a fermentation where we basically have been providing the cells with everything they need. So they are cultured in kind of a rich BSM medium. The pH is maintained at 5.5, and we're using ammonia as a base, which also supplies the cells continuously with nitrogen. So there are no limitations in this process. We get quite a high amount of biomass in this experiment, but also the productivity and the production of itaconic acid is quite high. So we get about 83.1 grams per liter. One important thing for downstream processing is that the broth that we provide is not very salty, or that the medium we use is basically has not a lot of excess components. And here in this fermentation, downstream processing has quite some troubles because our broth is quite salty, and it's difficult to extract the organic acid from this fermentation. So as a next step, we wanted on the one hand to reduce the biomass but also to use a medium that is better for downstream processing. So in F2, what has been changed is we have been implementing a nitrogen limitation. We have exchanged the ammonium base to potassium hydroxide so that our cells stop basically in cell growth, and that the carbon doesn't go into biomass, but rather into itaconic acid production. And what we can see is that with this strategy, it was possible to increase our product yield from 0.14 to 0.19, just by implementing this nitrogen limitation. And then in the next step in F3, a new medium was designed, a medium that is really minimized and limited in its salt content, so that it's easier for our downstream processing partners to handle our fermentation broths. And this exchange of the medium actually had not a big impact on our productivity. So we reach similar titers and also similar biomass formation. And then what we have also implemented to further decrease monovalent ions in our broth is that we have left out basically the pH setting in the batch medium. This is kind of a very small step. So normally when we prepare our batch medium, we put the pH with HCl to a certain value, and we didn't do that in F4. And it has no effect on the fermentation process. And we get again similar titers, and we were able to reduce the monovalent ions in our broth even further. So these optimization steps already helped a lot for downstream processing to make it easier to extract our organic acids. But what would be even better is to run our fermentations at a low pH. Normally we run our fermentations at pH 5.5, and here we have decided to decrease the pH to 3.5. This makes it basically with that change, our organic acid basically lies in acid form in our broth and not in salt form, and makes it even easier for our downstream processing partners to process our broths. But what we can see here in F5 is when we just decrease the pH and leave everything the same that we did before. So we have now this limited media. We have potassium hydroxide as a base. Our viability of the cells really suffer. The conditions of this low pH combined with these high itaconic acid concentrations, they take really a toll on our cells. And of course, the itaconic acid productivity went down, and the cells really had a hard time to cope. What Manya saw in her shake flask screenings was that when the cells are in a growing or in a metabolic active phase, they can handle these harsh conditions better. So what she performed in the next experiment is that she supplied ammonium shots during the fermentation. This basically, we don't have then this nitrogen limitation anymore. And the cells can cope a bit better with the conditions. So we could increase the titers from 23 to 36 grams per liters. And with that knowledge, it was actually decided that we switch the base potassium hydroxide back to ammonium, so that really the cells have again continuously supplied with nitrogen. And this really helped our cells in these harsh conditions a lot, reaching about 53.6 grams per liters of itaconic acid at pH 3.5. And then another minor improvement was made. The medium was again adjusted because, as I said before, we were working on a limited version of the medium to prevent excess components. However, this very strong limitation seemed to also have an effect on these low pH conditions. So here the minimal medium was adjusted again. Some of the salts were a bit increased so that our cells can cope even better. And with that optimization, we reach now 62.4 grams per liter of itaconic acid. We have a very low biomass, and we are able basically to run this fermentation at pH 3.5. So the outcome of this experiment here is really, really good. Yeah, just to summarize, what we have learned in these experiments is that the cells must be in kind of a metabolic active state to withstand these harsh conditions. And therefore, a nitrogen limitation is actually more counterproductive at this low pH. The biomass is anyways limited by the pH, so we don't really need the nitrogen limitation. And when we adjust this to our new process, we can reach high itaconic acid titers still. Yeah, and with that, I'm already at the end of my presentation. What we have learned in our research is that some organic acids have a higher potential on methanol than maybe others. So with lactic acid, we really encountered some regulations that we still don't understand quite fully, and that we need to research that we are able to be to make it possible to produce more lactic acid on methanol. Itaconic acid, on the other hand, works really good. And what we've also learned is that the process optimization is really unique to each situation. So when you want to ferment at low pH, you have to provide your cells with nitrogen. You make sure that there are no other stressors like nutrient limitations so that your cells get a bit more robust. Some of this data has been already published, so if you are interested, you can have a read. And I'm looking forward to your questions afterwards. With that, I want to thank you for your attention. And of course, I want to thank the Vivaldi team for their great contribution. Thank you.
Thank you very much, Simone. So questions will be answered at the end, as said. I think there is a lot of room for discussion. That's really, moving now into that area also of low pH fermentations, as Paul and Simone have addressed, is very important in the field. And we are moving away from the organic acid field now and from the methanol or CO2-based substrates to another waste stream, as said, and that's very important, I would say, to become able to recycle materials and get away from this end-of-life waste that our current economy is very much favoring. And I'm very happy to introduce Valeria Mappelli from University Milano Bicocca, talking about valorizing cotton waste by yeast fermentation. Valeria, please.
You're muted, Valeria. Yeah.
Yes, I think now you hear me.
Perfect.
Yeah, sorry. Thank you for inviting me to this very nice workshop. It's a pleasure to be here, and we're very interesting projects and talks. What I would like to talk today is, as Dita said, upcycling and valorization of cotton waste. And this is sorry, Okay, very graphical representation of what we do in our research group, which basically is to, the main aims is to produce either bulk or fine chemicals for very diverse applications that have an interest in different fields. It can be industrial field, material fields, or pharmaceutical and nutraceutical, using microorganisms by making use of their natural abilities or acquired abilities to produce those products. And to add a further level of challenge, what we'd like to do in most of the cases is to make the microorganisms not only producing these products, but producing them by consuming what is otherwise recognized as a waste, in a way that we would like to valorize at least part of what is nowadays only discarded as a waste. And one of these projects is in fact about valorizing the waste that comes from the cotton manufacturing. And why it's so interesting, it's because the nowadays economy of cotton, textile economy is fairly linear, meaning that from the manufacturing of cotton via different steps, basically to the final product. Eventually, the end of life of the product is most of the time the disposal in landfills. And how is the process of cotton textile manufacturing is described here in this scheme. And as you can see, in the different steps, you basically generate different kind of waste that are basically made of cotton scraps or wastewaters. And then you also have auxiliaries and dyes that enter the process to make the final product. And given the fact that the textile industry is basically the third biggest manufacturing industry in the world, and given the fact that fast fashion and fast luxury are increasing, also the volume of waste is increasing. And this results basically today in 73% of the world clothing being incinerated and landfilled. What about recycling? There are several applications that aim at recycling textile fibers, including cotton, and they have reached a fairly high technology readiness level. However, based on data, what we can say is that only a very minor percentage of those fibers are recycled. And if you think about cotton, less than 1% is nowadays recycled, and often results this recycling often results in a downcycling, meaning in the production of a lower quality product. So what would the aim of this project that I'm going to present is basically to add the circular, to make the textile industry circular, closing the loop in the textile industry, using the abilities of microorganisms and in particular of yeast to use the waste of cotton to generate compounds that can be reused in the textile industry, making then the textile production process circular. And we did this in collaboration with a cotton mill company based in northern Italy, which is Albini Group, which is producing textile cotton for clothes. And The case of cotton is particularly fortunate for microorganisms because from cotton, basically, you easily derive glucose because cotton is made, if we think about 100% cotton textile, this is made of around 92% of cellulose, which is a polymer of glucose. And how we managed to obtain glucose with very high, with a rather high yield from cotton is by combining first a mechanical pre-treatment of the cotton fibers that arrives as in this way, as you see from the cotton mill. In order to make these fibers accessible to the enzymes that are able then to degrade cellulose into glucose, we need a first step of chemical treatment that is meant to reduce the crystallinity of cotton, making the fibers accessible to the enzymes. And by treating the cotton with 50% sodium hydroxide, we managed to obtain glucose from this cotton waste with a yield of over 80%. We also, the aim we also had was to use a wastewater coming from the cotton mill, which was rich in sodium hydroxide, and in particular, in one of the processing steps that happens in the cotton mill, there is a wastewater coming that is made composed of 60% of sodium hydroxide. And to understand whether it would be possible to use such a lower concentration of sodium hydroxide and still keeping a very high yield of glucose from cellulose, we tested different percentages of lab-grade sodium hydroxide, noticing that a concentration between 10 and 20% would be still good to achieve the equal yield of glucose from cotton. And by using the actual wastewater from the cotton mill, we also reached a fairly high yield of glucose from cotton, meaning around 85% of yield of glucose from cotton. So this is what we basically did to scale up a bit the process in the lab. We wanted to make use of this of the glucose obtained from cellulose to make a fermentation, and this is what happened. So from cotton, this is what the pre-treated material that is being washed, and then the enzymatic hydrolysis was made happening directly in the fermenter at lab scale. And after 16 hours of enzymatic hydrolysis, we obtain a liquid medium rich of glucose, in particular with 75 grams per liter of glucose, where we could run our fermentation. So at this point, we were able eventually to use not only to make partial use, at least, to the cotton waste from the cotton mill and of the wastewater coming from the same cotton mill plant. With this glucose we obtained, we wanted to, of course, to upcycle via fermentation, and the first approach we had, the first aim, was to produce lactic acid. As it was said before in a previous presentation, lactic acid is very interesting as a platform chemical because it's used in the production of polymers, and is also interesting in the textile industry because it can substitute acetic acid that is typically used in the dyeing process. And what we obtain using the glucose coming from the cotton waste and using a Saccharomyces cerevisiae yeast engineered for the production of lactic acid at low pH, we obtained a yield of lactic acid from glucose equal to about 83%, which is a very good yield. And if we calculate the overall yield from cotton, we can say that 75% of cotton was upcycled to lactic acid. We still have a waste, but at least 75% of what would be a waste was now upcycled to lactic acid. And the further step we took was to produce an additional product from the glucose deriving from the cotton hydrolysis in order to be able to provide the cotton mill with a further product that could be used in their process. And this product is part of the natural colors that are used for dyeing cotton and wool fibers. And what we did was using Saccharomyces marxianus, so a different kind of yeast, and we compared to Saccharomyces cerevisiae, and the advantages of using this yeast are the thermotolerance, so it's more tolerating rather high temperatures. So this would allow eventually to develop a simultaneous saccharification and fermentation process, starting from the cotton hydrolysis and going directly to fermentation. And it's able to consume a wide range of substrates compared to Saccharomyces cerevisiae. And we would be able to use an engineered yeast that was already improved in the flux of the shikimate pathway, which was important for us for the production of natural colors, because we decided to produce colors that are in the family of the biolacine, and wool synthesis comes from tryptophan. And we basically used a strain of Saccharomyces marxianus, where we wanted to direct the tryptophan produced to the biosynthesis of violacines. And what you will see later is the production of PD-bio, by introducing into Saccharomyces marxianus these three genes, the CV-bio-A, B, and E, coming from Thromobacterium violaceum. The strain this shows the pathway, the natural pathway of Saccharomyces marxianus, where you can see that, as in most microorganisms, the amino acid biosynthesis pathways are mostly regulated by feedback inhibition. And the starting strain we engineered for the production of violacine was a strain that was optimized for the production of 2-phenyl ethanol, or in general of aromatic alcohols. And this strain was engineered basically to overexpress the genes that you see now written in green, in order to remove some of the feedback inhibition, and some of other genes were downregulated in order to reduce the flux diverting from 2-phenyl ethanol. And the strain was also engineered by overexpressing the genes in the PP pathway, in order to increase the level of PEP and phosphoenol pyruvate, in order to feed the pathway that would lead to the synthesis of tryptophan. And this strain had also this downregulation that was then resulting in a strain overproducing 2-phenyl ethanol, as you can see here. This strain was engineered in order to remove these properties and reduce the flux towards 2-phenyl ethanol, and this was successfully achieved. On the other hand, in this strain, we could not observe accumulation or the increase of flux of tryptophan. To improve the flux towards tryptophan, the gene where the feedback inhibition was removed was expressed, but on the other hand, also in these cases, no tryptophan could be detected as produced in this strain, and in order to see an overproduction of tryptophan that would be useful for the production of violacein, we had to reduce or downregulate the levels of PHA2 gene, and only in this case we managed to actually increase and detect the synthesis of tryptophan. So the chassis strain for the production of violacein was built, and now the pathway for the synthesis of violacein was introduced, and in particular, we managed to obtain a Saccharomyces marxianus strain, KM Bio 100D strain, able to produce prodeoxyviolacein, and possibly also the dimer, which is the oxychromatoviridans. This is what we obtained in fermentation, and as you can see in this video, I don't know if it's very visible, but you see now and then something dark passing in the medium, and these are basically patches of the colors that we managed to produce with the Saccharomyces marxianus in a batch fermentation, still at lab scale, where then the color was then given to the cotton mill and used to dye the cotton and wood fiber. And what we can say is that the color, the fibers were successfully dyed using the typical process that is implemented in the cotton mill. There are some aspects that still need to be improved, but the good news is that we basically made use of the wastewater of the cotton mill, and we made use of the waste scraps they produced to eventually produce both lactic acid and colors that can be used in their dyeing process. So adding, let's say, in this way, managing to close the loop of a typically linear economy using industrial microbiology, and in particular, and synthetic biology to engineer Saccharomyces cerevisiae and Saccharomyces marxianus strains. With this, I conclude by thanking, of course, the coordinator of the project, which is Professor Paola Branduardi, and the two PhD students that were mainly working on this project, that are Pietro Butti and Marta Simonetti, who was working at the cotton mill and in our lab. And I thank you for your attention.
Thank you very much. So yeah, we have time for questions now. Thanks for all the speakers, also for your perfect time management. I see that there is a couple of questions, and nicely also distributed to the different talks. So let's get into it. The first one goes to Pau, and it's about the increase of biomass when co-feeding formic acid. Have you conducted any techno-economic analysis on this strategy? Do you believe the increase in biomass is sufficient to offset the cost of formic acid?
Okay, so the direct answer is no, not yet. I would say we haven't performed a techno-economic analysis. We started doing some, but we have in VIVALDI consortium, we have a partner currently doing LCA, so life cycle analysis. They did it for itaconic acid, I believe, if I'm correct. Okay, they've done it, and they started recently with 3HP. But for the economical part, not yet. We were aiming to do it, but let's say, more advanced stage where we have more data, particularly in fed batch cultures with formic feeding. What would be interesting is not to increase 20% yield on biomass, but to increase 20% of yield of the product. Okay, so minimizing product biomass formation and while increasing product formation. There could be an option there, I believe, if we manage to increase to minimize biomass and then optimize while increasing product metrics. Okay, but I think it's too early for that. Still, I don't know.
Okay, thank you. Yeah. The next question goes to Simone, and it's about the transporters. So yeah, you showed the MFS transporter for itaconic acid that was overexpressed. And the question is whether you also overexpressed the lactic acid transporter.
Yes, yeah. So we have been investigating a monocarboxylic symporter. It was Yen1 in Komagataella phaffii for lactic acid export, but it didn't really had an effect, unfortunately. So we couldn't increase the titers with this transporter. And as we have seen then that the product rather affects the transcriptional regulation of our genes, we didn't continue with searching for other transporters, I have to say. So we haven't tested more transporters in that regard, yeah.
Right, thank you. Yeah, actually, it's the Yen1 is the one that has been also described for Saccharomyces cerevisiae lactic acid producers to be functional or to improve production. Okay, thank you. Then we have two questions to Valeria. The first one, are there some ideas to use plants to clean the wastewater? For example, Rhabdomyces reed. So that goes a bit off the yeast fermentation topic. I mean, yeah, this plant wastewater treatment is an important topic, of course. Do you have any information in that regard?
No, I don't know anything much about the plant. I mean, to treat the wastewaters in this case. I know there are, I think, some electrochemical fermentation or kind of fermentation, by electrochemical systems that can do by something to, reduce or oxidize the molecules. But I don't have, to the best of my knowledge, I don't know whether these have been applied in this case to degrade colorants.
Okay, thank you. Yeah, then the next one also to you, Valeria, it's regarding violacin production, if it's secreted from the cells or intracellular, and if intracellular, if you did use the biomass to dye the cotton, or
No,
it's when secreted, probably.
Yeah, it's secreted, and we didn't use the biomass to dye the color, but only the broth, the cultural broth, basically. Yeah.
Okay. I have one more question to you about you showed cotton fibers and like the pure cotton that is, as you say, quite pure cellulose. What about mixed fibers? I mean, it's an issue that more and more of the textiles on the market are mixed fibers of like cotton or other cellulose-based and synthetic fibers.
Yeah.
Is that
this is basic. Yeah, sorry.
No, go ahead. You know where I'm
Yeah.
heading to.
No, this is basically the big challenge in recycle or upcycling or recycling the final products, because these are mostly made of mixed fibers. In fact, I mean, what we were focusing now on what is the pre-consumer waste, because we use the waste from the cotton mill, where basically the fibers were not mixed yet, and in that case, basically they produce a textile which is made of pure cotton. But and even in that case, when we hydrolyzed the cotton, there were still some residual that was not degraded, meaning that the cellulose is not 100%. I mean, cotton is not 100% cellulose. So those debris, let's say impurities, were kept in the fermentation, and it was fine enough. But yes, since we didn't let's say cope with the difficulties to work with mixed fibers, as we focused on these cotton mill wastes. But yes, the mixed fibers is the biggest challenge in this case. Yeah.
Would it be that probably, let's say, the non-cotton residues would remain as solids after hydrolysis, so they could go into other recycling streams?
It could, yeah. It could also. It could be an application. Yes, of course, one approach. Yeah.
Okay. Yeah. Thank you. Just to say, also the speakers, of course, have the option to ask questions.
I have one. I have one for Simone. Simone, you show very nice results at pH 3.5 with the strains that really are performing, sustaining growth and production until, I think, more than 100 hours, which is quite a long time. Did you check viability by flow cytometry, or how is it?
Yeah, we checked the viability, and at low pH it's really reduced. So it goes down to 10% almost. Sometimes even further. And what we could see is that if you supply them with nitrogen and also nutrients in the medium, they perform better. But when we run these fermentations for such a long time, there is a time point where then they just start dying. So it's getting too much. And what we now continue to do is we want to do adaptive laboratory evolution with these strains. To make them a bit more robust at these harsh conditions, because they really suffer. They perform and they still produce, but there is a point where they can't bear it, or they have a hard time to bear it.
Okay, thank you.
Thanks. There is one, let's say, a more general question in the Q&A, whether the consortium is still open for collaborations. Let's say, to enter the consortium, the quick answer is no, unfortunately. First, of course, these funding schemes are closed as they are funded. And second, the funding period will end mid of this year. But I think collaborations are certainly interesting for future, and I would hand over to Albert, actually, who is the coordinator and is coordinating us so wonderfully, and also the expert in the wastewater area. If you have any addition to that.
Yes, no, as you said, now the project is almost finishing, but if you want to send me an email, we can explore novel collaborations for sure. And since I have the word, I want to take the opportunity to tell everyone to save the date of May 5th. We are doing the final event of Vivaldi. It's going to be streamed online. It's a couple of hours event where we will explain all the achievements that we've done, not only in the field of yeast-based yeast fermentation, but also on CO2 capture and bioreactor chemical ammonia recovery. So I hope you are interested, and you will see the performance of a great consortium during the last 4 years. So thanks for giving me the opportunity to do my advertisement.
Of course, and that's of course very important also to spread the word on the next events.
Yeah,
So,
maybe to add
yeah.
there, sorry, Dieter, to stay up to date with all the news about the project, how you will be able to access the final conference, don't hesitate to visit our website. I will post now in the comments the link, and there you can find where to join us on LinkedIn, on X. You can subscribe to the newsletter on the bottom of the homepage of the website, so you will be able to know all this information. Also, the recording of this webinar will be available either today or tomorrow, and it will be posted through these channels, so keep an eye on that.
Okay, this is already like near to closing words. I'd say there is still a chance for one or two final questions, if there are. If not, let me thank very much our three speakers for super interesting talks. I think we have all seen that there is really a lot of opportunities in yeast biotechnology and using yeast fermentation for circular bioeconomy. And I mean, VIVALDI is one great project in that direction, but of course a lot more is going on in the different partner labs and via the VIVALDI webpage, as Lara said. You find all the information and you will see what else we are doing and what is moving forward in several directions. And with this, thanks a lot to the speakers. Thanks a lot to all of you who have participated, and a big thank first to Albert to coordinate that project so wonderfully, and a huge thanks to Lara for not only organizing this webinar, but organizing us all the past years in our outreach activities, and being great encouragement always, and super great support for all of these activities.
Thank you
Thanks.
for all the content that you're bringing. It's super easy then to share such amazing results.
Great. Yeah. So this has been really, really wonderful. And huge thanks again to Valeria for joining us. I think that has been wonderful also, to step beyond our, let's say, thematic limits and really show that there is much more out there that we can do, and that's hopefully like getting the way into industrial applications. Good. Then,
Okay. Yeah, thank you.